Hello, welcome back to Topic 1 Cell Biology GCSE. Today we are going to be going over binary fission. So binary fission. Prokaryotic cells can reproduce using a type of simple cell diffusion called binary fission. In binary fission, the cell splits into two. So the circular DNA and the plasmids replicate. The cell gets bigger and the circular strands move to opposite ends of the cell. The cytoplasm begins to divide and new cell walls begin to form. And the cytoplasm divides and two daughter cells are produced. Each daughter cell has one copy of the circular DNA, but can have variable number of copies of the plasmids. So here we have an image showing that. So in step 1, we have the circular DNA, which is shown here, replicating. So we go from 1 to 2. In step 3, the cytoplasm begins to divide, and a new cell wall begins to form. In step 4, the cytoplasm divides and two daughter cells are produced. Each daughter cell has one copy of the circular DNA, which we can see here, but it can have a variable number of copies of the plasmids. So here we have 4 plasmids and here we only have 2. Prokaryotic cells replicate by binary fission. Bacteria can divide very quickly if given the right conditions. The right conditions include a warm environment and lots of nutrients. Some bacteria, such as E. coli, can take as little as 20 minutes to replicate in the right environment. However, if conditions become unfavourable, the cells will stop dividing and eventually begin to die. So we can use mean division time to find the number of bacteria in a population. So the mean division time is just the average amount of time it takes for one bacterial cell to divide into 2. If you know the mean division time of a cell, you can calculate how many times a cell has divided in a certain time, and so the number of cells it produces in that time. So an example of this, so we have a bacterial cell that has a mean division time of 30 minutes. How many cells will it have produced after 2.5 hours? So as we know, the mean division time is the average amount of time it takes for one bacterial cell to divide into 2. So after 30 minutes, we'll have 2 cells. After an hour, we'll have 4 cells. So we have 2.5 hours and we want to put this into minutes. So to get into minutes, we multiply by 60, and this is 150 minutes. And we want it in minutes because the mean division time is given to us in minutes. So now, to find out the number of cells, we do 150 minutes divided by 30 minutes. And this shows us that the cell has divided 5 times. We have 5 divisions. So to calculate how many actual cells we have, we do 2 to the power of 5. This is because at each division, for each cell, we have 2 more cells, and that equals 32 cells. Thank you for watching, and don't forget to subscribe.